All right, let's look at a few practice questions. So the first one, what are the two primary neurotransmitters of the efferent branch of the peripheral nervous system and which neurons release them? Okay, so our two neurotransmitters, remember, are acetylcholine on the parasympathetic side or norepinephrine / epinephrine on the sympathetic side. Okay, let's see here. Excellent, so I'm seeing in the chat here, yes, epinephrine and norepinephrine, that's one division, but then we also want to add acetylcholine, which is on the parasympathetic side, okay? So, right train of thought there. Number two, what is the neuroeffector junction? This is the junction between the postganglionic neuron and the effector tissue. And it's different and separate from the neuron to neuron synapse, which is at the autonomic ganglia. It's also different from the neuromuscular junction, which we will speak about in our next unit. The neuromuscular junction is where we have a neuron synapsing on to skeletal muscle. Remember, we're not talking about skeletal muscle here. The autonomic nervous system does not control skeletal muscle. These are muscles that we do not have voluntary control of, like the smooth muscle that's in your GI, the smooth muscle that's in your blood vessels, smooth muscle of your uterus, smooth muscle of your bladder, smooth muscle that is in other areas of the body, respiratory tract, cardiac muscle in your heart. These are not muscles that we control. Okay, so that is what sets the neuroeffector junction apart from things like the neuromuscular junction, as well as a neuron to neuron synapse. And then number three, in what regard does it differ from an ordinary neuron to neuron synapse? In which respects are they the same? So we kind of just touched on that there a little bit. They're the same in that they are, they're facilitating synaptic transmission, right? We've got the voltage-gated calcium channels. We've got the neurotransmitter being stored in the vesicle. It's similar in that regard, in the general sense of the events, the receptor on the adjacent end to receive that neurotransmitter. So those are some ways that they are the same. They are different in that there's no axon terminal. So at the neuroeffector junction, instead of having a neuron to a neuron with two discrete axon terminals, we see the nerve endings of that postganglionic neuron, and then we see the effector tissue that is being synapsed, onto. That's kind of one difference. And then the other two main differences was the idea that we have the varicosities instead of the axon terminal, and then that the cleft is a little bit wider. So it's more of a distance for that neurotransmitter to cross. Okay. And again, when we get to these checkpoint questions, make sure even if you don't respond and share your answers, make sure that you write the answers down. It's going to be really helpful for you when you go back and try to study this, that you have succinct answers that are answering the questions, and you can guide yourself in terms of preparing for the exam and so on. All right. We'll get into our third objective here in the last few minutes, which is to describe how the central nervous system regulates the dual innervation of the autonomic branch of the peripheral nervous system. In other words, how do those integrating centers receive those input signals, make sense of everything, and then send that response down to these effector tissues? Okay. So we talked about this idea that there's a dual innervation happening to all of our organs. The heart, for example, it's receiving parasympathetic input mainly via the vagus nerve, right, cranial nerve 10. It's receiving sympathetic input, right, coming from T1 to L2 there. So it is receiving input from both of these divisions. And so while I'm at rest, let's say I'm just sitting here resting, neither of them are overexerting the other. There's this dual innervation happening that's keeping me at rest. If it's time for me to run from a threat or flee or even exercise, I'm going to have the sympathetic slightly outweigh the parasympathetic. And if I'm sleeping or in a deeply rested state, I'm going to have the parasympathetic, again, more outweighing the sympathetic. But in order to have, normal everyday function, both of these divisions are, sending input to all of our organs to keep things at homeostasis. If we see an increase in the parasympathetic system. It's Thanksgiving. I've had a big dinner, large turkey dinner or whatever. I'm resting, I'm digesting. I'm going to see an increase in the parasympathetic input, but then likewise, in order to have coordination there, I'm also going to see a decrease in the sympathetic input as well. The opposite is true. If I'm running from a threat, running from a bear, I'm going to see an increase in my sympathetic activity, and then that's going to be coupled with a decrease in my parasympathetic activity in order for that change to be effective. It makes no sense to increase the sympathetic and still have parasympathetic active, right? That's going to be sort of counterintuitive or counterproductive. So there's that comparative secrecy, whereas increasing one causes a decrease in the other, and the opposite is true. Let's talk about an example here of how all of this comes into play. So we've got someone who stands up really quickly, and whether you know this or whether you're conscious of this or not, when you stand up really quickly, the force of gravity pulls all of your blood to your lower extremities, and your upper extremities actually go through a period of decreased oxygenation, decreased vascular flow. So what your systemic arteries, your core arteries, your upper limb arteries, what they detect is a drop in blood pressure, right? That's kind of abnormal. They will then pick that message up on those visceral receptors that the blood pressure has decreased. They're going to send a signal on an afferent fiber, afferent pathway to the central nervous system. And again, correlating what we said here about action potentials and about communicating different signals, the frequency of the action potential is going to decrease. So thinking about that frequency coding that we talked about in the nervous system, the way that the nervous system communicates a change in a signal, a decrease in a signal, is to decrease the number of action potentials that are firing per minute. So the way that we send that signal of a drop in blood pressure to the brain is dropping the frequency of the action potentials on that afferent fiber. The cardiovascular centers in the brain will pick that up. They'll detect that we have a drop in blood pressure, and they will respond accordingly. They will decide via the integration of those integrating centers that we need to increase the heart rate in order to correct that drop in blood pressure. So they're going to have to send two signals according to that comparative secrecy. They're going to send a signal to increase the sympathetic activity to the heart. But likewise, in tandem, they're also going to have to send a signal to decrease the parasympathetic activity, which will increase the heart rate and prevent that slowing down influence which the parasympathetic signal is sending. That is what will increase the heart rate, and that is what will bring the blood pressure up and correct our initial error signal. Alrighty? Okay. So this is just an example of how both divisions are working together, incorporating the autonomic nervous system, incorporating the central nervous system, the integrating centers, in order to bring about and maintain homeostasis.